And we are joined in the studio by Dr. Carlie Lalone, scientist at the EPA. Thank you so much for being here, Dr. Lalone. So you're researching chemical safety. I'd like to know historically about animal testing. What has animal testing looked like to inform hazard and risk assessment?
Yeah, well, thank you for having me. And so chemical safety, typically for hazard and risk assessment, has relied on whole organism toxicity testing. And so that's looking at measurable, observable toxic effects typically. So that would be something like reproduction, growth, mortality. And more and more so people were using model organisms. So that means that there's certain test species that tell us the most about other species. So considering human health risk assessment, a lot of times we'd utilize mammalian model organisms to tell us about potential effects. And in the context of ecological risk assessment, we'd utilize various species that represented different taxonomic groups to be able to tell us something about the diversity of species in the environment. Now, typically these tests are ran, and then we get estimates of toxicity, and then people would apply safety factors to account for interspecies differences. And we've recognized over time that these whole organism toxicity testing, though it's been very valuable, there are more efficient ways that we can screen and understand chemical toxicity. And so there's been efforts to be able to reduce the number of animals used in toxicity testing. So certainly there's an international effort to be able to understand how we can use different types of approaches where we can be more efficient, both as far as cost and time that goes towards toxicity testing, as well as, again, reduce the number of animals used in toxicity testing.
Well, in fact, that leads me to my next question, and that is why and how can we get away from animal testing?
Right. So, in 2007, the National Research Council actually had laid out a vision and strategy for toxicity testing in the 21st century. And now, over 10 years later, we've really started to realize that we can utilize more and more in vitro or cell-based assays and computational approaches, as well as omics technologies such as genomics, proteomics, metabolomics, to understand toxicity and toxicity across species. And so this is important. Much of this work is ongoing. There's a lot of efforts to develop what are termed new approach methods. So that's kind of an umbrella term that's used to categorize all that type of new approaches that are being developed to be able to test organisms, but more on a cell-based or computational tools to be able to think about biological pathways.
What do you hope people take away? What is your main goal for your session today?
Yes, so the session today is focused on cross-species extrapolation and really thinking about animal alternatives. And so we'd like to have discussions focused on what are regulatory needs for cross-species extrapolation, and then what are current pipelines that risk assessors, decision makers are using that they think that they could incorporate some of these types of methods, and what are the challenges. So having those types of discussions help people like me that are developing these types of tools to be able to understand what the data needs are, what the challenges are, so that as we're developing these types of approaches, we can have those considerations. So then from the researcher's perspective, we're learning from the decision makers, the regulators, what their needs are, but then also it's an opportunity to introduce these types of approaches that may be more of a black box to people. They might not understand or have a full understanding of their capabilities, but through training and this type of outreach and these types of discussions, we're able to have communication and bring people into these types of sciences and how they can help in cross-species extrapolation for regulatory decision making.
Sounds like there's a lot of work to be done.
Yes, absolutely.
Thank you so much for being here and
Yes.
giving us such a thorough explanation. Dr. Carlie Lalone with the U.S. Environmental Protection Agency.